السلام عليكم ورحمة الله تعالى وبركاته. I'm Salman al-Mahdi, and in this third lecture of lipid metabolism, we are going to discuss ketogenesis. Our objectives are to identify the ketone bodies and their function, to define ketogenesis and state when and where ketogenesis occurs, describe the liver's role in ketogenesis, and describe the synthesis of ketone bodies from acetyl-CoA, and then to describe ketone bodies metabolism in their target tissues, with emphasis on the role of 6-amino-CoA transferase, and to describe the consequences of excessive ketone body reduction and explain the relationship between fatty acid oxidation and ketogenesis. So what are ketone bodies? Ketone bodies are water-soluble polar molecules that are produced in the liver during the breakdown of fatty acids and ketogenic amino acids. They function as an alternative energy source. Alternative to what? To glucose primarily. When are they needed as alternative energy source? In conditions of prolonged fasting, carbohydrate restriction, and uncontrolled diabetes. And in all these examples, there is increased lipolysis and increased free fatty acids oxidation. They serve as a fuel for extrahepatic tissues, such as the brain, heart, and renal cortex. The brain particularly cannot oxidize fatty acids, so when there is restriction of carbohydrate in the diet, or when there is diabetes and insulin is not there to enhance glucose entry into the cells, other tissues may shift to fatty acids as a fuel and to be oxidized. However, the brain cannot oxidize fatty acid. 50% of its energy demand will be covered by ketone bodies, so they are important source of energy for the brain. What are the ketone bodies? There are three, including acetoacetate, 3-hydroxybutyrate, and acetone. This is acetoacetate, 4-carbon atom compound, acetone, and 3-hydroxybutyrate. Ketone bodies are interrelated. Acetoacetate, for example, acetoacetate undergoes continuous spontaneous decarboxylation to form acetone. And acetoacetate and 3-hydroxybutyrate are interconverted by a 3-hydroxybutyrate dehydrogenase, and their equilibrium depends on the ratio of NAD to NADH. And in the blood, the ratio of 3-hydroxybutyrate to acetoacetate may be equal, that is 1 to 1, or it may be 10 to 1 in favor of 3-hydroxybutyrate. The normal concentration of ketone bodies in the blood does not exceed 0.2 millimole per liter in a well-fed animal. We specified here well-fed state to say that carbohydrate is abundant, glucose is present, so there will be low levels of lipolysis and free fatty acid, low concentration of free fatty acids in the circulation, low oxidation, and low ketone bodies. The least rate or amount of ketone bodies will be present in a well-fed state. It is a normal state, and in that state, ketone bodies in the blood does not exceed 0.2 millimole per liter. And since they are polar and water soluble, so they can be excreted in urine. And normally, in a human, ketone bodies concentration in a 24-hour urine sample does not exceed 1 milligram. Maximal utilization of ketone bodies is at 12 millimole per liter. At that concentration, the highest level of utilization of ketone bodies by the body occurs, after which ketone bodies will be accumulated because they have already saturated as enzyme systems that normally use them. And please note that 3-hydroxybutyrate is the predominant ketone body in both blood and urine. So when we measure or when we test urine for ketone bodies, or when we analyze a blood sample, we are actually looking for the concentration of 3-hydroxybutyrate. Ketogenesis, as the name implies, ketogenesis, synthesis or genesis of ketone bodies. It is a biochemical process whereby ketone bodies are formed. Ketogenesis is associated with fatty acid oxidation in the sense that metabolic conditions that are associated with high rate of fatty acid oxidation are also associated with increased production of ketone bodies. And tissue-wise, as we said, liver synthesizes ketone bodies. In fact, liver is the only organ in humans that has significant quantities of ketone bodies to the blood. And this applies not only to humans, but to all non-ruminants. In ruminants, ketogenesis takes place in the epithelium. Within the cell, ketogenesis takes place within the mitochondria. The flow of ketone bodies refers to the transport or movement of ketone bodies from the site of production, that is the liver, to the site of utilization, that is the extrahepatic tissues, brain, renal cortex, and heart. And the net flow of ketone bodies is from the liver to the extrahepatic tissues in a one-way street or direction. Why? Because active enzymatic mechanisms for the production of ketone bodies in the liver is coupled with the very low activity of enzymes responsible for utilization. So the liver, although it synthesizes ketone bodies, it does not use them as energy source. And the reverse occurs in extrahepatic tissues. Extrahepatic tissues doesn't have the ability to synthesize ketone bodies. However, they use it as a source of energy, so it will be mainly for utilization, while the liver will be mainly for production. So the movement will be from the liver to the extrahepatic tissue across or along a concentration gradient from the very high concentration as a site of production to very low concentration as a site of utilization. As we said, liver synthesizes ketone bodies from acetyl-CoA. It will be carried in the blood to the extrahepatic tissues to be oxidized in citric acid cycle to produce energy. Excess ketone bodies will be spilled in urine, or acetone will be volatilized through the lungs. And your question came earlier. Why does the body synthesize ketone bodies as alternative energy source instead of directly using acetyl-CoA? So, if you go back here, the liver used acetyl-CoA that is coming from breakdown of fatty acids and acyl-CoA to produce ketone bodies. Ketone bodies then will be used to regenerate acetyl-CoA. So why not use acetyl-CoA directly as a source of energy? Why do we need ketone bodies as an intermediate? And you need to state at least 5 justification for this fact. The reactions of ketogenesis are 4, starting with formation of acetyl-CoA, and then formation of HMG-CoA, splitting of HMG-CoA, and finally formation of beta-hydroxybutyrate. The first reaction, which is formation of acetyl-CoA, can be done in one of 2 ways. Either this acetoacetyl-CoA, a 4 carboatom molecule, is the end product, the final end for carbons of oxidizing an acyl-CoA, fatty acid, by beta oxidation. So the last 4 molecules will be acetoacetyl-CoA. This acetoacetyl-CoA is the end product of fatty oxidation, beta oxidation of a fatty acid. Or this acetoacetyl-CoA can be formed from condensation or combination of two molecules of acetyl-CoA by sialase by reversal of a sialase reaction of beta oxidation. So we can use the final force reaction of beta oxidation, sialase or sialytic lipid, to combine acetyl-CoA with another molecule to combine acetyl-CoA with another molecule of acetyl-CoA to produce acetone acetyl-CoA. And then this acetyl-CoA will be used to form a molecule that is called 3-HMG-CoA or 3-hydroxy 3-methyl-butyryl-CoA. 3-HMG-CoA is an intermediate that is present here in ketogenesis, and we are going to come across it again when we talk about cholesterol synthesis. So acetyl-CoA will condense with another molecule of acetyl-CoA to form the 6-carbon atom CMG-CoA by NHMG-CoA synthesis. One CoA is released. We have two acetyl-CoA molecules here and one acetyl-CoA here. So the formation of 3-hydroxy-3-methyl-butyryl-CoA required 3 molecules of acetyl-CoA. This HMG-CoA will be split by HMG-CoA lyase into acetyl-CoA and acetoacetate. Then 3-hydroxybutyrate will be formed by 3-hydroxybutyrate dehydrogenase. How? By, as we said, acetoacetate is convertible with 3-hydroxybutyrate using NADH. It should be converted or oxidized to NAD. So now we have our ketone bodies. It's hydroxybutyrate can diffuse in the blood to be used as an energy source. So how does the target tissue or the extrahepatic tissue is going to use this ketone body? Ketone bodies are oxidized proportionately to their concentration in the blood. The higher the concentration of ketone bodies in the blood, the higher rate of oxidation of ketone bodies as energy. And as we said, the cutoff point is the 12 millimole concentration, after which the oxidative machinery will be saturated. Acetoacetate and 3-hydroxybutyrate can both be readily oxidized by extrahepatic tissues. However, acetone cannot be oxidized easily in vivo. It is volatilized in the blood, so it will be lost in breath with expiration. Utilization is via two reactions. The first one is activation of acetoacetate. So this acetoacetate has to be activated by adding a CoA molecule, and this is a cytosolic process whereby acetoacetate reacts with succinyl CoA. This is on the circulating free fatty acids. Oxidation by beta oxidation, formation of acetyl-CoA. Acetyl-CoA then will be used to form HMG-CoA, then acetoacetate, then 3-hydroxybutyrate. This 3-hydroxybutyrate will diffuse to be used in an extrahepatic tissue. This hydroxybutyrate will deform acetoacetate because the reaction can be reversed, and acetoacetate will react here with succinyl CoA to form succinate, to release succinate and form acetoacetyl-CoA. This CoA came from succinyl CoA, and the reaction is catalyzed by succinyl-CoA transferase, transferring a molecule of CoA from succinyl-CoA to acetoacetate. Acetoacetyl-CoA is now active. It can be split by a sialase to reform acetyl-CoA, and this acetyl-CoA can be used for energy to be oxidized by acetic acid cycle. How ketogenesis is regulated? Regulation of ketogenesis takes place at 3 crucial steps: at the level of mobilization of free fatty acids from the adipose tissue, and at 2 partitions. The first partition is between esterification and oxidation, and the second partition is between ketogenesis and the acetic acid cycle. Let's see how does that. Okay, so we are talking about a first level of regulation It is the level of lipolysis, the mobilization of the fat stored as triacylglycerols to form fatty acid. Here is the first level of ketogenesis regulation occur. A second level of ketogenesis regulation is at the partition between oxidation and esterification. So we have our free fatty acid has entered the liver as acyl CoA. This acyl CoA is either goes through beta oxidation or esterification. This decision is anti-regulation or a site of regulation, particularly through the carnitine palmitoyl transferase 1 enzyme. And then after acetyl-CoA is formed, it also has to decide whether to go through ketogenesis or citric acid cycle for complete oxidation. This level here is also a site of regulation ketogenesis. Regarding the first step or site of regulation, mobilization of free fatty acid from the adipose tissue, this step is inhibited by insulin. Insulin inhibits lipolysis, while glucagon, the insulin antagonist, promotes lipolysis. The rate of free fatty acid production by adipose tissue regulates the rate, or the concentration of free fatty acids in the plasma, and the concentration of free fatty acids in turn determines the free fatty acid uptake by other tissues. In particular, the liver. The liver extracts up to 30% of the free fatty acids passing through the liver. So almost a third of the quantity of free fatty acids that passes through the liver will be taken up, and this is an evidence to the central role of the liver in metabolism in general and in fatty acid metabolism and ketogenesis in particular. The second level of, as we said, of regulation is at partition of esterification and oxidation. After uptake of free fatty acid by the liver, the free fatty acid has two fates: either oxidation by beta oxidation or ketogenesis, or esterification to triacylglycerols and phospholipid. The control, as we said, is specifically at the level of the carnitine palmitoyl transferase 1. Esterification to triacylglycerol and phospholipid is not rate limiting. The third is the partition between ketogenesis and citric acid cycle. The acetyl-CoA formed by beta oxidation is either oxidized further, as we said in citric acid cycle, or used for ketogenesis. As the level of serum free fatty acid increases, the concentration of free fatty acid in blood or in serum increases, more acetyl-CoA will be converted to ketone bodies and less is oxidized by the citric acid cycle. So the level of the concentration of free fatty acid in the blood, the higher it is, the more ketone bodies are produced and the less oxidation of acetyl-CoA by citric acid cycle. Regarding the biomedical relevance of ketogenesis, we need to go through three terms, and these are ketonemia, which is abnormally high quantity of ketone bodies in the blood; ketonuria, which is an abnormal high quantity of ketone bodies in urine; and ketosis, which is an overall condition that includes both ketonemia and ketonuria. Acetoacetate and 3-hydroxybutyrate are moderate strong acids, and their continued production eventually exceeds the body's buffering capacity, leading to ketoacidosis, which is a form of metabolic acidosis. In starvation, there is normally increased rate of ketogenesis, and we do have ketosis. However, this is a simple form of ketosis. It is not very high, and it doesn't have complications. It serves a function of providing energy, as we said, for the extrahepatic tissues, particularly the brain. However, in disease conditions such as uncontrolled diabetes mellitus or poorly controlled diabetes mellitus, a fatal form of ketoacidosis can occur. And we are going to discuss a case that illustrates this fact. A 14-year-old girl was admitted to the emergency room in coma. So she was not conscious. She was well until 2 weeks prior to admission, when she developed sore throat, moderate fever, loss of appetite, and poor feeding. So here we have a background of infection. Loss of appetite, that means feeding is less. It is not a well-fed state. It is going perhaps into starvation, restricted intake. You expect that carbohydrate and glucose are low, the fatty acids are now increasing in the blood, and there is additionally there is loss of appetite and poor feeding. So there is no replacement or replenishing of the nutrients, particularly carbohydrate. Few days prior to admission, she developed polydipsia, which is excessive thirst, polyuria, which is increased urine frequency and volume even at night. Polydipsia and polyuria are both features of diabetes, as you know, and we are specifying here at night because usually the number, the frequency and volume of urination by day is much more than night, and there is an acceptable number of of urination, after which this is labeled as excessive. On the day of admission, she did vomited and became drowsy. So now the condition is progressive. There is vomiting, there is aggravation of dehydration, and there is deterioration of consciousness. On examination, when this patient was examined, she couldn't be aroused, so she was in coma. She was dehydrated, her skin was cold, and her breathing was in deep manner. It is called cold breathing with fruity odor. The fruity odor here signifies the presence of acetone in the breast. Acetone is a volatile ketone body. She was hypotensive, her blood pressure was going over 60, and she had fast tachycardia. Her pulse rate was very much increased at 115 beats per minute. The laboratory finding is interesting. Look at the glucose here. Blood glucose concentration was 35 millimole per liter. The normal range states that the upper limit should be at 6, and she has 35 millimole per liter, very much increased. Beta hydroxybutyrate is 13. It should be less than 0.25. Acetoacetate level was 2.8, should be less than 0.2. Bicarbonate was 5, it should be 24 to 28. So she has low level of bicarbonate. We're going to discuss in a little bit why bicarbonate is low. pH, as you can see here, it is more in the acidic spectrum. Why? Because of the presence of the acids, the ketone bodies. Ketone bodies are acids. Urea is 12, high. Creatine is 160, also high. Potassium is high at 5.8. The urine contains glucose. Normally it shouldn't contain glucose. Ketone bodies were present at 4 Normally they should be nil or trace. The diagnosis was a type 1 diabetes mellitus presenting with diabetic ketoacidosis. Ketoacidosis meaning this is an acidosis that is due to ketone bodies in the context of diabetes. Here the insulin deficiency is the cause of main cause of the condition. Initial management should include fluid replacement. As we said, she is dehydrated. Should include insulin therapy, correction of the electrolyte imbalances, and addressing the underlying infection. Biochemical interpretation here, the polyuria, the polydipsia, hyperglycemia, and glucosuria are all features of diabetes. Ketonemia, ketonuria, as we said, overall term is ketosis. The acidity, the low pH, the low bicarbonate means it is a type of metabolic acidosis with respiratory compensation. Increased urea in the creatinine or renal impairment due to dehydration and protein degradation. The increase in potassium concentration is due to lower uptake, and the lowered uptake is due to the insulin deficiency itself. The pathophysiology of DKA is deficiency of insulin and relative excess in the antagonistic hormones such as glucagon. And the clinical picture of DKA reflects metabolic abnormalities of carbohydrates, of lipids, of proteins, as well as abnormalities of pH and electrolytes, all of which are due to the insulin deficiency. How is that? Diminished synthesis of insulin by the beta cells have multiple effects. For example, in carbohydrate metabolism, there will be decreased glucose uptake by certain tissues, and the increased glycogenolysis and increased gluconeogenesis, all of which will manifest as high glucose in the blood hyperglycemia. The effect on lipid metabolism will be increased lipolysis, increased fatty acid oxidation, and increased parallel production of ketone bodies, leading to increased both free fatty acids and ketone bodies in the blood. And the effect of insulin deficiency on protein metabolism will lead to decreased protein synthesis and increased metabolism, eventually leading to increased creatinine. Potassium, water, and pH are affected in the sense that there is decreased entry of potassium into the cells because insulin is not there. There is water loss secondary to glucosuria because here this is a form of osmotic diuresis whereby glucose, when it is exiting the body, takes or water follows it. Acidosis, of course, due to the increased concentration of ketone bodies, they are acids. So the overall manifestations will be hyperkalemia, acidosis, and dehydration. This is all. Thank you very much. See you next time.